really glad to be here to talk to you about the human microbiome, especially in relationship to what happens to the microbiome, the ways that we're changing it. And I hope to show you that, as Susan mentioned, that there may be some important consequences. Here's a map of Greenland, two maps of Greenland in 1992 and 2002. And we all recognize this as evidence for global warming, for climate change. Now, actually, based on the election yesterday, there are some people who may not recognize that, but here in New York, we know it's true. So we can think of this as a change in our macroecology. And what I want to talk to you is about a change in our microecology that I believe is on the same scale. So let's begin at the beginning. What we know is that ever since there were animals on this planet, they have had microbes living in them, residential microbes. This goes back 500 million years, maybe a billion years. The microbes don't live just anywhere. They live in particular niches. What that means is that the microbe on your arm is different, for example, than the microbe in your belly button. We know that some of the organisms are persistent. That means that we acquire them early in life, and they may persist weeks, months, years, decades, for our entire lifetime. And there are others that are transient, short-termers. Some of the microbes are conserved. That means that there are microbes that every person in this room shares, and there are microbes that are host-specific. What that means is that it is likely that even with this large number of people, every one of you has some microbes that no one else in this room has. So that creates a very interesting tension. Most of the cells in the human body are not this red leg, the human cells, but they're the microbial cells. And even more so, most of the genes in our body are microbial. Human genome, 23,000 genes. Human microbiome, on average, about 2 million unique genes, somewhere around 99%. So we can ask a series of biological questions about the microbiome. What is the identity of the microbes that populate their host? Who are they? What are they doing? What's their function? How is the host responding to them immunologically? What are the forces that maintain equilibrium among the populations? Why don't they kill us? Why don't we kill them? How can we live together for so many years? We're particularly interested in that question. What are the unique characteristics of each individual? How do we differ from one another microbiologically? And most importantly, how can we manipulate this? How can we advance the science in this field so that we can understand the answers to those previous questions so that we can improve health and fight disease? When babies are born, their microbiome is very unadult-like, and then gradually and progressively, it becomes more and more adult-like until it is. And what's important is that the age of this transition is the age of 3 years. So this first 3 years of life is the time when the microbiome is developing. It's the most dynamic. It's the most different from adult. And of course, this first 3 years is the time that babies are developing also. They're developing their metabolism, they're developing immunity, cognition. How long has this been going on? This is a phylogeny. This is a family tree of these wild hominids, and you can see there's a certain pattern of branching. And this is what we accept is the ancestry of the hominids. And here is a family tree of the microbes, of the fecal microbiota in these same animals. And if you look carefully at the branchings, you see that they're all the same. So this property, this congruence between the two family trees, means that the transmission of the microbes, just like the host genes, the transmission is vertical. Just like their human or their monkey genes, they get their microbial genes. So this tells us it's inherited, it's from mom, and it's also evidence for coevolution. It's been going on this family tree of hominids is about 4 million years of evolution. It begins at the moment of birth. As the baby passes from the womb through the birth canal, they acquire their mom's microbes. We all, by and large, have acquired our mom's microbes. As we go out the birth canal, we're covered by them. We swallow the microbes. Then the baby comes into contact with the mom's skin and the mom's breast, and the mom is kissing and licking the baby. And this is the intergenerational transfer of microbes. For us mammals, it's been going on for 100 million years. And it begins starting at the moment of birth. That's how it's been. But now, in these modern times, moms aren't the same as they used to be. They live in an environment with antisepsis, antibiotics that are selecting different organisms in them. Their diets are different than they used to be, with different antibacterial materials in the diets, and other factors. And babies aren't the same as they used to be. In fact, today in the United States, 32% of babies are born by cesarean section. That's 1 in 3. In some places, like Rio de Janeiro and urban China, it's about 50%. So this is a huge change in the beginning of the microbiota, and then the babies are washed extensively, very early. They are given formula, which resembles human milk in color, but not in many other ways. And very importantly, babies are getting a lot of antibiotics, and I'm going to talk about that. So this whole early period of transition, the intergenerational handoff, is very much impacted. And I want to introduce a concept that we've been developing over the last more than a decade, which I call the disappearing microbiota hypothesis. And it has 4 tenets. Beginning in the late 19th century, changing human ecology has dramatically altered the transmission and maintenance of our indigenous microbiota. These changes in ecology have affected its composition, and we have examples of this. The altered composition affects human physiology and thus disease risk. We have some examples of this. And very importantly, loss of ancestral bacteria usually acquired early in life is especially important because it affects a developmentally critical stage. So this is the hypothesis. About 5 years ago, Dr. Stanley Falco at Stanford and I extended it, and our view was back in the old days, whenever that was, women had a normal ancestral microbiota. But if through the course of life she happened to lose organisms, but it could not be restored horizontally from other people, then the next generation would be born with less of that microbial ancestry. And so on and so forth over generations. And so our view is that, in fact, the changes are cumulative across generations, that it has been stepping down, that we humans are losing biodiversity, especially in developed countries like the United States. Recently, I found a study in Japan that supported this. This was a study of an organism that used to be the major dominant organism in the human stomach called Helicobacter pylori. And this was from a study of 3 generations in Japanese families. And here were the grandmothers, here are the mothers, and here are the children. So we have evidence with this organism for this kind of projected step down. So what are the consequences of this? Well, I want to talk for most of the time about obesity. Because as all of you know, obesity has become an enormous problem in the United States and, in fact, around the world. So this slide looks at obesity trends in U.S. adults. These maps are from the Centers for Disease Control. And in 1989, there is no state in the United States where more than 14% of adults are obese. By 2010, there is no state in which fewer than 20% of adults are obese, and the national average is about 30%. Unfortunately, this also looks like the election map. Now, what's remarkable to me is that it's happening everywhere, and not just in the United States, it's happening all over the world. And the other point is that there's only 21 years between these two maps. Something extremely powerful must be going on to produce a change of this scale, something very widespread. This is the percent of obesity in kids of different ages. And the point is, it's going up. Even in the youngest kids, it's going up. And this is representative of a large body of data that the signals from obesity are beginning early in childhood, just when the microbiota is developing. As indicated from the title of the talk, or of the invitation, I'm very interested in antibiotics because antibiotics were discovered in the 1920s, the 1930s. They first began to be used in the 1940s and have been used widely ever since. And so the timing of the introduction of antibiotics fits with the development of obesity and some other diseases as well. So let's look at some of the data on antibiotic use. This slide looks at the top 8 prescriptions for children in the United States in 2010. And of the top 8, 5 of them are antibiotics. And if you add these numbers of courses of antibiotics, it's more than 40 million. More than 40 million courses of antibiotics in our children, basically every year. And that's just for the top 5. Now, in fact, antibiotics are available everywhere. In the United States, you need a prescription to get antibiotics. But in many countries of the world, antibiotics are available on demand. This slide is from a small village in the Amazon. This is a picture I took with my phone. In between the drinks and the batteries were the generic over-the-counter antibiotics. In China, it is estimated that antibiotic use, which does not require a prescription or a physician, is 2 to 4 times as common as ours. It's higher, we believe, in India as well, but we just don't have the numbers. So wherever our civilization has gone, so have antibiotics, because they are important life-saving drugs when they are used for life-threatening illnesses. Now, recently, the Center for Disease Control did a survey of antibiotic use in the United States, and what they found is that there were 258 million courses of antibiotics used in the United States. That's 833 courses per 1,000 population, or 5 courses for every 6 people. And according to the CDC investigators, this has been going on year after year. Now, the antibiotic use isn't uniform by age. Kids in the first 2 years of life have the highest rate of use. It goes down, and then it comes up as people get older. The average child in the United States in the first 2 years of life is getting almost 3 courses of antibiotics. And by the time they're 10, they've had 10 courses. And by the time they're 20, they've had 17 courses. Again, this is across everybody. Some kids have more, some have less. But these are big numbers. And in fact, these numbers are consistent with many other surveys, albeit those are much smaller surveys than this huge nationwide survey. Now, recently, a colleague of mine, Dr. Jeff Gerber at the Children's Hospital of Philadelphia, shared with me data that's soon to be published, looking at variation in use of antibiotics, of prescribing of antibiotics by doctors associated with CHOP, which is actually one of the best children's hospitals in the United States. They looked at 29 pediatric practices associated with CHOP, 350,000 child visits, and they found between the least prescribing and the top prescribing practice, there's a 100% difference. There's a 2-fold difference between top and bottom in the rates of prescribing antibiotics. And when they look at prescribing of broad-spectrum antibiotics, there is a 4-fold difference. So all doctors aren't the same. There's a lot of variation in antibiotic prescribing. Now, if we go back to the CDC data and ask about the geography of antibiotic use, we find some interesting trends. The national rate is 833 per thousand. In the Northeast, it's 830. In the Midwest, it's 868. In the West, it's 638, much lower than the national average. And in the South, it's 936. It's much higher than the national average. And in fact, between the South and the West, there is a 50% difference in the per capita rate of antibiotic use. And as a specialist in infectious diseases, I can tell you that there is not a 50% difference in the rate of serious bacterial infections. This difference reflects practice and culture. Now, what I found especially interesting was to compare the two maps, the CDC map in 2010 of obesity and the map of antibiotic use. You will see that there's a lot of resemblance here. Enormous resemblance, strikingly non-random. Now, these are observational data. They don't tell us whether A causes B, B causes A, or C causes A and B. But they certainly are consistent with the idea. So because these were observational data, we decided that we were going to try to do some experiments to better understand the relationship between antibiotics and obesity. And we decided to study animals. For the last 70 years, farmers have been feeding antibiotics to farm animals, to cows, to their livestock, as growth promoters, to make them bigger, to bring them to market earlier. The farmers use very low doses of antibiotics, what we call subtherapeutic antibiotic treatments, or we abbreviate as STAT, and they use them for growth promotion of their livestock. They have shown that they can fatten up their chickens, cows, pigs, sheep, just about all the livestock that they try. And this is a big swath of vertebrate evolution. It's not just mammals, it includes birds. Over the years, the farm scientists have investigated a broad class of agents, and they find that virtually every antibacterial agent they try works regardless of the chemical structure of the antibacterial, its class, its target, or its bacterial spectrum. Antivirals don't work, antifungals don't work, specifically antibacterials. And the earlier the antibiotics are started, the bigger the effect on growth rate and on feed efficiency. This is the ability to convert food calories into body mass. And you want that if you're a farmer, but maybe not if you're a parent. So we began a series of studies in mice in which we gave mice stat levels of antibiotics or not. We examined their characteristics, which we call phenotypes. We analyzed their microbiome, and we looked for relationships. So we've been working at this for a while, and I want to show you the results of about 5 studies. The first study was led by Dr. Il-Sung Cho when he was a fellow in my lab. He's now a member of the faculty at NYU. And Il-Sung gave in their drinking water mice 4 different courses of antibiotics at levels that are used on the farm that the FDA approves. And here you can see these big increases in body fat percentage, and we can illustrate this by what's called a DEXA scan, where the fat is colorized in yellow. You can see these examples. We postulate that the antibiotics are mediating differential selection of the microbes in the colon. And that's what antibiotics do. They select. They kill some organisms, they suppress others, and that allows certain bacteria to thrive. That's natural selection. We showed in Il-Sung's study that the antibiotic dosing changed the composition, the population structure of the bacteria. It altered the representation of genes involved in the synthesis of short-chain fatty acids, which the bacteria and we use for energy. We showed that there was increased synthesis or production of the short-chain fatty acids, and this goes by the portal blood circulation to the liver, and we showed that it induced genes in the liver to create fat. And then we showed that the mice were fatter. So on the basis of these studies, Laurie Cox, who was a graduate student in the lab, did a series of experiments. I'm going to show you a few of them. And the first experiment I'm going to show you is an experiment that we called FatStat, where we asked what's the effect of combining a diet that's high in fat, it's high in calories, what we call a high-fat diet, or HFD, and stat on body composition. So Laurie began her experiment with two groups of mice, either getting penicillin in their drinking water, that's the stat group, or not, that's the control group. Everybody got normal mouse food for the first 17 weeks of life, and then half of the mice were put on a high-fat diet, and half of the mice remained on a normal diet. We looked at male mice and female mice. We looked at the total mass, how big they got, their fat mass, and their lean mass, how much muscle. Here, this black line is the group that got normal chow and no antibiotics. That's the control group. And the group that got antibiotics were bigger, just like happens on the farm. And if they were put on a high-fat diet, they got bigger still. And if they were on a high-fat diet plus antibiotics, they were the biggest. So there's an additive effect. When we look at muscle mass, we see that they put on more muscle, just like the animals on the farm. And when we look at fat mass, we see the big accumulation in fat happens in the mice started on the high-fat diet here. There's an additive effect. So up to this point, the mice had been getting antibiotics for all their life. But people generally don't get antibiotics for their whole life. They get shorter courses of antibiotics. So next, Laurie did an experiment that we called Durastat. She asked, is the adiposity, is the fatness durable with only a limited antibiotic exposure? So in addition to the no antibiotic group and continuous lifelong antibiotics, she had a group of mice that got only 8 weeks of antibiotics or only 4 weeks. All three groups of mice receiving antibiotics were bigger in total, lean, and fat mass. So 4 weeks was enough for the full effect. They didn't need to be on it for their whole life. And in other studies, Laurie showed that that first 4 weeks of life was much more important. The effect was much stronger than if they got it later. So now, what about the microbiome? In this form of research, in microbiome research, our holy grail is a fecal transplant, or some kind of transplant, where we can test what happens if we give the microbes to a germ-free animal, an animal that doesn't have its own microbes. So Laurie did an experiment that we called Transtat. She asked, is the growth phenotype, is the characteristic of enhanced growth transferable by the microbiota alone, forgetting about the antibiotic? So for this experiment, her donor mice were mice that either received the antibiotic or not. She picked mice from the middle of the group, so not to bias things. She sacrificed those mice and preserved the microbes in their cecum in a special transport medium that preserved the viability of the organisms. And then she gave them by an oral gavage, or she administered it into the stomach of recipient mice. And these were germ-free mice. Here they are in their bubble, received from Taconic Farms, Swiss Webster mice. They were 3 weeks old. And now these mice are conventional. And then they were housed and kept for the next 5 weeks. So the mice that received the microbiota from the stat mice were increased in total mass. No change in lean, but an increase in fat. So from this study, we have evidence that the microbiota alone are sufficient to transfer the characteristic. Now, I want to finish the experiments just by telling you one other line of work, and that has to do with diabetes, type 1 diabetes. Some of you know, and that's juvenile diabetes, some of you know that this disease, which is a terrible disease, which forces children to start taking insulin and is associated with long-term disease. There's no cure for juvenile diabetes, and people, on average, do not have a full lifespan. Type 1 diabetes is increasing around the world. On average, in developed countries, it's doubling about every 20 years. And so Alexander Livanos, another student in the lab, decided that she was going to see if there could be any relationship between this and antibiotic use. So she worked in another model of mice, a model called the NOD mouse. And these are mice that spontaneously develop something that looks just like type 1 diabetes. And Ali's hypothesis was that early life antibiotics would change the gut microbiome and lead to type 1 diabetes accelerated in NOD mice by affecting the expression of genes in the small intestine and by changing the T cells in the intestine. And to make a long story short, Ali has shown all of these, and recently she successfully defended her PhD, so she's now Dr. Livanos. And unfortunately for me, she's graduated, which is good news for her, but we hate to lose her. So her work has shown that even in this system, not just obesity, but type 1 diabetes, and we think it's because of the changes in the T cell, have accelerated. So I've been telling you about studies in mice. What about humans? So there have been a number of epidemiologic studies that have looked at the relationship of antibiotics and other early life perturbations. And now I'm going to tell you about two studies that we did with colleagues in England. In the early 1990s, a big cohort of more than 10,000 children was enrolled in England in Avon. It's called the Avon Longitudinal Study of Parents and Children. Lots of data were obtained. They were followed for the next 15 years. And Leo Tresandi in the team asked, is there a relationship between infant antibiotic exposures and early life body mass? And the main result of the study is children who were exposed to antibiotics during the first 6 months of life were more likely to have markers of obesity. And now there have been 4 new studies that have shown the same phenomena. In Colorado, here in New York, Noel Mueller has done work, and also in Manitoba. And in another study from Allspac, we've looked at the association of cesarean delivery with child adiposity from age 6 weeks to 15 years. Jan Bluestein led this study, and the results are the same. The kids who were born by C-section were more likely to have markers of increased adiposity when you control for all other known factors. Also, there have been 4 studies that have shown the same phenomena. And so how do we put it all together? So when babies are born, they aren't fully formed. And we know in the uterus, babies have stem cells. And stem cells are cells that are pluripotential. They have to decide how many times to go around. And if they're a mesenchymal stem cell, they have to decide, should they become fat or bone or muscle? So we've been paying a lot of attention to diet and calories. But our view is that we need to start paying attention to the early life microbes and the cells they're talking to, because the hypothesis is that these commingled populations are sending signals out that are affecting these developmental decisions. And here's a cartoon of the new days in which kids have perturbed early life microbes due to antibiotics and other factors. They have different microbes. They have different cells that are signaling, creating a different context. So what do we need to do? In the scientific community, we need to do research about antibiotic consequences to find out, are antibiotics as benign as we think? Are they as free as we think? And if we find there are risks, we have to be better educated. As scientists, we have to develop more specific agents. Instead of broad-spectrum antibiotics, we have to develop narrow-spectrum so we can treat infections more precisely. That's going to mean better diagnostics. We need to remediate. After somebody takes an antibiotic, we need to replace some of the organisms that are lost, but we don't fully know what they are yet. And from that will come probiotics, not necessarily the ones you're buying today, but scientifically developed probiotics. We need to enhance some of the organisms that are depleted. They're present but depleted with prebiotics. And maybe we have to do a reversal. We have to start giving back some of those vanished organisms, replace them maybe with isolates from the Amazon or from Africa. and ultimately we have to monitor. Now, I want to go back to the antibiotic use in the United States from that CDC study showing this cumulative use that I showed you before, because shortly after this was published in the US, a group of scientists from Sweden said, let's look at our data. And they found something quite remarkable. By the age of 3, the average Swedish child has about 1.4 courses of antibiotic, where our child has about 4. And when our child has 10 antibiotics by 10, they only have 4. At every age, the Swedes are using only 40% of the antibiotics we're using. So it shows, and the Swedes, by the way, are at least as healthy as we are. So it shows, at least by this metric, that about 60% of the antibiotics we're using, at least, are unnecessary. So there's lots of room for improvement. So what's the doctor of the future going to be doing? I think that the doctors, especially for kids, in addition to the usual well baby checkup, they're going to analyze their poop. And they're going to figure out which of the globally necessary microbes they need and which of the personal microbes they need, and they're going to administer them, and then we're going to assess. And that way we're going to get the kids on the best path for lifelong health. It's going to take some science to get there. And I want to thank all my colleagues at NYU, including Isabel Teitler, who's here tonight. So thank you very much for your attention.